So there are many times in paleontology where you're going to come across something and it's going to be so strange that you don't know what to do with it. And so when you encounter something new, something unexpected, how do you go about maybe not figuring out what it is, but figuring out what it could be and figuring out how it works? Because I think that's ultimately the more useful, productive enterprise. So here is an example of just that, and pretty much anybody who's working in the Cambrian will have encountered something a little bit like this, because these are from the Burgess Shale, where you have excellent preservation, and you have soft part preservation, and you have no hard part preservation, but you have little echoes of where the hard parts used to be. So this is Cyphosoctum gregarium. Here is a modern reconstruction of it. And just look at that. Just look at it. That's pretty cool looking. It appears to be an animal. So this is located in a zone and at a time before there are anything like multicellular plants per se. So yeah, animal of some sort. And it has a stalk, and the stalk has what looks like a holdfast at the bottom. So presumably these are all in contact with the bottom, with the mud, potentially anchored in the mud or onto whatever they can just get their little holdfasts hooked onto. Inside this little cup, you have a bunch of these sort of fronds, and each frond has little sort of mini, it's almost like tentacles coming off of it, like what you'd picture in a baleen whale. Like, so already I think we're thinking maybe a filter feeder of some kind, and that these are potentially the filters. And we'd want to look at the symmetry, we'd want to look at a lot of things. But the first thing to notice is the stalk, and there's an outer stalk and an inner stalk, and they fossilize really differently, which kind of suggests that they were made of something different. And they hypothesize that the inner stalk here is actually the hard stuff that's giving it some rigidity, and the outer stalk is more of a casing and is keeping that part of the organism alive, presumably. Okay, here is another one. So generally, when dealing with something new, it helps if you can find more than one. And if you can find more than one in more than one state of preservation or just state of repose, like the way that it is laying there, if it's different, you can see a different side of the animal. So this one has been completely squished down, and you can see all of these little sort of sideways tentacles coming off here. You've got a cup down here at the bottom, to which you might be thinking, hey, it could be echinoderm. These could be little feeding arms. Maybe it didn't figure out fivefold symmetry yet. We definitely know that the early members of that group had not found fivefold symmetry or any symmetry at all, at least in the early points. But yeah, it's hard to say. Or rather, bilateral symmetry, but in the wrong direction. Like they're a pancake, but they're bilaterally symmetric in this direction. Almost, yeah. But there you can see the holdfast a little bit clearer. And here we've got sort of the tentacles, and we've got those magnified. And so you can see the potential tentacles, which we're now probably thinking are going to be for feeding, because there's a lot there. I guess you could also imagine respiration as a potential use for these, because you need a lot of surface area there. Generally, these organisms are going to devote a lot of their body parts and time and effort into feeding foremost, and then having babies second most. And as part of feeding, I think oxygen exchange is going to be a big part of it, especially if, as we think, these lived in a semi-anoxic environment, like with very little oxygen. They might have to go to greater lengths in order to get enough oxygen to keep their metabolism going. Or they may have adapted for life in an area and a metabolism in an area with very low oxygen. Because it's not zero. You could still do a little bit of oxidative respiration, but yeah, it wouldn't be easy. Next thing you can do is, if you've got a three-dimensional specimen, and why not, you could dissect it. Why not? And so they're dissecting this one, and look in the center, there's this great big, sort of like a cone shaped structure, sort of flattened out, but so it looks like a wedge. You've got this dark material here, which is actually going to be similar to what you see down here in the inner stalk area. So we don't know if those are made of the same substance or if those are attracting the same kinds of bacteria during fossilization. You could be thinking, well, is this like the mouth parts and the gut runs down the stalk? Because there are some early stalked creatures that do have their gut down in their stalk, essentially. Yeah, and some modern ones. Yeah, it happens. Okay, another good thing to do is to establish a size range where you just get the smallest one to the biggest one. You lay them out next to each other, or you take pictures of them all and lay the pictures out next to each other, because this is the digital age. We're not animals. And here you can see a little tiny one next to quite a big one. And yeah, you can compare the parts. And so here we've got this little thing right in here about where you would expect a mouth or maybe the part of the gut to be. We've got a little inner stalk running inside of an outer stalk, so that's fine. And the feeding arms are not as developed in the small one. Those appear to really elaborate and develop later, which may reflect greater oxygen requirements. You know, you'd grow more lungs so you can do more respiration, so you can be bigger, so you can then catch more food, so that you can grow more arms, so you can use more oxygen, so you can get bigger, etc. Round and round and round. Yeah. Hey, blue whales are a thing. Whatever. Here is a sort of a deeper dive. They're looking at, they found another one that was squished in a very weird way, where one of the feeding arms came out, and you can see these little tentacles coming off the side there, and they're zooming in on the holdfast. In this one, it doesn't have much of an inner stalk, or it's not very differentiated, but the holdfast is kind of differentiated. It's coming out in this darker black. So it's maybe attracting more of those iron pyrite producing bacteria, if this was the original pyritization that then kind of went away. Yeah, these fossils have been through enough diagenesis that it's hard to be certain what they were originally, and it's even difficult to reconstruct the past sometimes. Here they found one where it's facing straight up, so it's basically it was laying like that and got crushed, like the one up here. If you got crushed from this angle and just sort of smashed and flattened out, here you can see that central disc that may be the digestive tissue, may not, but yeah, you can see that. And so now you can actually get a feel for the symmetry, because all the other specimens have been flattened, and they're flattened sideways, and so some arms are on top of others, and so you can't see the whole thing unless you dissect it. And by dissecting it, you're going to destroy whatever's on top, and these are very precious, so they don't want to do that. But they've got enough of them, and so here they can actually spot, and yeah, sure enough, there are 1, 2, 3, 4, 5, 6 little arms. So 6, that number gives you pause, because if it's a bilaterian, 2 would be fine. If it's an echinoderm, we would expect maybe 5. That would be a good number. Or there are some that have 3, some that have twofold symmetry, some that don't really have symmetry. But it doesn't. If it was a ctenophore, like a comb jelly, we could say, hey, maybe let's look for 8 or some multiple of 8. But no, 6 is not a multiple of 8. It's 75% of 8. So that's not going to do it. Yeah. What the heck? Crazy Cyphus acutum gregarium? What's going on here? And look at that, yeah, little potential digestive organ in there in so many of these. Yeah, that's interesting. So yeah, you find them from different orientations. You want to try and get a feel for the symmetry there. Sometimes the symmetry gives you a real strong clue. Other times it will give you no clue at all, like in this case. So we've got a 6-fold kind of symmetric as far as we can tell thing so far. Probably an animal, definitely with a stalk. Here they're going to zoom in. They're going to look at that holdfast. They're going to look at the inner and outer stalk. And it almost looks like that's just like a coating that's on there. See how it sort of stands out, it sort of bulges out from the side? That could be like a layer of slime or something like that, or it could have its own special little wrapper around it. But yeah, clearly the inner and outer stalks are very distinct. And then the holdfast appears to project from the outer stalk at the bottom and sort of fan out, at which point it would hold on to stuff, presumably. There we go. All right, so what are you left with? The people in this paper describe it as something new. They suggest some possibilities, like maybe an echinoderm that didn't figure out, hadn't learned to count to 5, but it could count to 6 already. Later workers have suggested that this is part of that group, maybe, of the ctenophores. The ctenophores that had a sort of mineralized projection that's stuck down into the ground. And that these are actual comb rows that are on the inside and are being used for feeding. I mean, it could be, that's for sure. But is it? They don't know, and they're not my fossils. I certainly don't know. But we've built up a picture of an organism. We've got a mode of feeding. We've actually learned a lot about it. We know that there's a 2-component stem down in here, and it's all just nice observations. So now let's go a little more. So we've reconstructed the organism. Well, how do they how do they work? Like with the Fractophusis, which you don't remember these names, but that was the ediacaran organism where you have the big one in the center, and then it puts out little stolons, and it grows 4 smaller ones around it, and then each of them grows 4 little mini ones around that, and each one of those grows 4 little teeny tiny ones around it. So you end up with individuals of the same size in a particular position relative to something else. And so here are some individuals of different sizes, shorter and longer stalks, which may suggest that the stalk is contractile because the head is about the same size here, or is that the stalk can grow independently of the head. Also a possibility, because it's hard to rule a lot of things out with a fossil like this. Got a lot of shape variation, and I think that's a pretty good clue that you're dealing with something that is kind of soft, because either its shape is varying during life or it is easily deformed upon crushing death from above, which, a lot of us are easily deformed by landslides, by undersea landslides. There's a little more on the shape variation. This one is showing that there's actually some of these, sort of sides come together at the tip, and the others, they're kind of apart. So it's like, could this thing open up a little bit? There aren't any where it's really wide open, except for that one that was smashed straight down from the top like that. And so that one's probably not reliable. But the ones on their side that maybe didn't have to be open, mostly they're shut or ever so slightly open, which could allow water to come into the chamber if they're going to do filter feeding. And so here's our internal anatomy, and we can see all those same parts that we saw before, but in more detail than ever. You've got your different parts here. They're actually describing that as the stomach down in here. And it's got sort of a middle and a lower chamber of that. And then there's a further chamber that's underneath that, where presumably digestion is going to occur. And those digestive tracts in Chengjiang fossils, for example, usually end up a different color, potentially from the different pH and the different bacteria that attracted, which led to different diagenesis, which led to different color in the eventual fossil that resulted. The next thing they do, which maybe should be the first thing that you do with your fossils, is measure. And so they make measurements of lots of different ones, measurements of all the different parts. Like here they are. And I think maybe what they should have done, and probably did do somewhere else, was instead of just saying like the length of the stem is 31.6 plus or minus 12.5, and we measured 177, I think some of these may work best as relative length if they can show that the size of certain components scale with each other. Like when this part gets bigger, this other part gets bigger. Like on your hand, your fingers grew at a relatively predictable rate. Baby hands don't look completely different from adult hands. They don't have like two super long fingers and just little claws down here. Yeah, because that would be weird. Yeah. But since they studied all the different life stages, they can actually make these conclusions. This part I like. They excavated these things in situ. So it's on a big field. It was one event that killed all of these things. It was like the whole family was living together. And they preserved the orientation of these. So they got the relative orientation of these slabs, probably because they saw a lot of the individuals lying in the same direction. And so it turns out that most of them are pointed this way, if like 360 degrees. So at that particular angle, which they think is the angle that sludge running downhill on that slope would be sort of pushing them. So they're probably sort of flattened in this way. And then some of them got kind of tumbled and appeared in other directions. But yeah, the definite directionality in three different big assemblages of this same creature from nearby. And so it was a similar type of event coming from a similar direction, which, landslide and the slope above is the conjecture. Yeah. And then once you have the measurements, you can start to measure and compare orientations. You can compare sizes. You can, this is looking at population variation on different slabs. And so you got your other loose ones over here. And you got some populations that are mostly kind of older and mostly kind of larger. You got some populations that are mostly younger, maybe like a little nursery that's just getting started, and a bunch of kind of middle-aged populations, just assuming that the bigger a thing is, the older it probably is. Yeah, which is how most things work. And then they go to pour just more cold water on you, me, and everything by saying, hey, look, relax. You can't call these things stem group cnidarians or ctenophores or echinoderms with any kind of confidence. They may not be any of those things. Here are examples of some other animals from the Burgess Shale that actually fall into groups that kind of make sense, and this is what we know about them. So here is a stalked holothurian, probably. This is a stalked sea cucumber relative, we think. Yeah. And so those do not normally have a stalk, but this one for sure does. And so that's a very weird thing. You've got actual stalked echinoderms where you have a little bit of preservation here of what looks like an actual calyx. That's great. You've got some other tentacled creatures here that can apparently open and sort of fan out their little tentacles or pull them back together, as you can see here. And I don't believe we know exactly what those are. They're just a stalked animal from the Burgess Shale, like the figure says. So, did we come to a satisfactory conclusion? No. Yeah. Paleontology is not about conclusions. It's about the process, of course. It's about doing the research, putting together the data, and trying to understand. Yeah. And at best, we'll have more of an understanding. And of course, calling these one group as opposed to another basically has no meaning when we're dealing with things as far down into the crown group as these are in the crown group of metazoan animals. So, yeah, they are part of the Burgess Shale. They were stalked. They probably were filter feeders of some sort because it doesn't look like they could move around to be very good predators. And they do have these little sort of tentacles with side tentacles on them with a lot of surface area on there. So it would make sense if they were able to do that. But yeah, beyond that, their affinities remain uncertain. And at some point, hopefully, we find more relatives that are intermediate, for example, between these and something else that's a little easier to interpret. And if not, they're just part of the big, beautiful, wonderful world of the Middle Cambrian as preserved in the Burgess Shale. So, thanks very much. I hope that was interesting.